This lab is called the MOL: Conversions, Mass Determination, and Hydration. The lab is pretty simple. You're going to go ahead and first, in part 1, find the masses of certain pure substances. Now, they're not exactly pure, but they're close enough to pure that the error is going to be less than 1% of the total mass. So we can go ahead and assume they are pure substances. Remember, in any experiment, there's always going to be some experimental error because of the experimental design. Not because of what you did, but because of the way the experiment's designed. Although these are over 99% pure, they're not pure. They're not 100% pure. So you have to keep in mind that all experimental data is going to have some margin of error. And that's called experimental error. It's part of the experimental design. We're going to find the number of moles in one of those aluminum cups. We're going to find the number of moles in your glass rod. And we're going to find the number of moles in your cotton ball. Now you have to look very carefully. The aluminum is pure aluminum. It's an element. The glass rod is made out of silicon dioxide. It's actually just sand. It's glass. Silicon dioxide. And the cotton ball is a polymer of glucose, cellulose. And when glucose polymerizes, which means you take one glucose molecule, you attach it to another glucose molecule, it attaches to another glucose molecule, and you get a long string of glucose molecules. And another example of a polymer is like DNA, that's put together of different bases. Or protein, which is a string of amino acids. Many things are polymers. And cellulose is a polymer of the sugar glucose. However, when it polymerizes, to make that attachment between the two glucose molecules, we have to dehydrate the glucose. So though glucose is C6H12O6, in the cellulose, it's C6H10O5 because a water is removed to allow the two glucose molecules to attach to each other. So keeping in mind the molecular structure of each glucose molecule in the cellulose, we can go ahead and find the mass of the cotton and convert it into moles of glucose, keeping in mind that the glucose molecules are a little bit different than a C6H12O6 glucose. In other words, you're going to use glucose in the form C6H10O5 because that's the polymerized version of each individual molecule to find the moles of glucose in your sample. The second part of the lab is going to take alum. The chemical name for alum is aluminum potassium sulfate. This is something called a hydrate. When it sits, and many chemicals are hydrates, when it sits in air, it will attach to waters from the atmosphere from anywhere. And so alum is actually a hydrate. It has some number of waters attached to the molecule. Our goal is to find the waters of hydration. How many water molecules are attached to each alum molecule? In order to do that, we're going to take a sample of alum hydrate. Alum hydrate is when the water is attached. When that water is attached, it has a certain molecular mass, which we don't know because we don't know how many waters are with each alum particle. We are going to take the alum, take the total mass of the sample. We're going to heat it, which makes the waters go away. It dehydrates. It turns into anhydrous alum. Then it's just the aluminum potassium sulfate and no waters attached. We can find its dry mass after it's anhydrous, find the moles of aluminum potassium sulfate. We can then figure out the difference between the anhydrous form and the starting form, how much water went away when we heated it. That is our mass of water. We can find out how many moles of water there are then. So now we know the moles of alum, anhydrous, and we know the water. We can make a ratio of the water to the anhydrous alum to find out how many moles of water are for each mole of aluminum potassium sulfate or the alum. That's the goal of the second part of the lab. Follow the directions carefully. It's pretty straightforward. You're going to have to use a balance. Keep in mind when you use a balance, we are going to use all of the digits, just like any other measuring device. Even if it's a 0 at the end, which people seem to like to not use, write down the 0. Always write all of the values from every measurement that you can. And remember, usually if we're measuring with a device, we'll measure all of them that are marked on the device and we'll estimate one more. The balance does the estimating for you. So that last digit in the mass you get is going to be estimated. Do not ever drop that digit. It's an important, significant figure which contributes to the precision of our calculations and our measurements. And that's this lab. Once again, where we're going to take masses, find the moles by doing some calculations, and finding the mole ratio of an anhydrous. It's kind of like an empirical formula type thing. The moles of water to the moles of anhydrous alum, or aluminum potassium sulfate.